Hey guys, welcome to Proactive Prudence. In this video, we'll be studying about microbiology and the very first topic, which is history of microbiology. So, what is microbiology according to you? Yes, microbiology, as the term says, micro meaning small, which cannot be seen from your naked eye. Since we know what is microbiology by splitting those terms, now we'll see what is the definition of microbiology. So, the definition of microbiology is it is a science of living organisms that are visible under microscope only. So, you have under the microscope, whichever living organism that you observe is considered to be the study under the branch of science as microbiology. So, what is medical microbiology? Now we have done with microbiology. Now we'll see what is medical microbiology. We'll read up the definition first. It deals with the causative agents of infectious diseases of man, his reaction to such infections, the ways in which they produce diseases, and the methods for their diagnosis. So, what is medical biology? Microbiology is it will deal with the organisms or the microbes which cause diseases in man. The first point is it will cause diseases in man, and the reaction that is being given from the human body from the infection is called as is the second one. And the third one is you are going to find out the ways in which the diseases are being produced from the microorganism. And fourthly, you'll see what are the different methods to diagnose the infection. So firstly, what it was is the causative agent which causes infection in the man. That is called as infectious microbe. The second point is the reaction which humans shows to the infection or the microbial invasion to the human body. And thirdly, it is the different ways the disease is being produced when the infection has happened. And the fourth point is the methods for the diagnosis. How are you going to find out that this infection has been caused? So that method is called as diagnosis. So that is called as medical microbiology. So two scientists named Vero and Columella have postulated that the diseases which are caused by invisible microbes, they are called as Animalia minuta. Before the term microbe came into use, it was called as Animalia minuta by Vero and Columella. So, what is Animalia minuta? It is an invisible organism which causes disease in the human body or the any other animal. Now, moving on, we have some more scientists who have contributed in the field of microbiology. In here, we have Fracastus of Verona, a scientist from Verona who proposed the term contagium vivium, that is, the living organisms which have been causing the infectious diseases, that is called as contagium vivium. Moving on, we have Antony van Leeuwenhoek, the famous scientist who had his major contribution in forming the microscope. So, what is his contribution? We'll see what Anton Leo Van Hook was. A draper of Delft, Holland, whose hobby was grinding, credited for observation and description of bacteria. So when the very first contributions made by the scientists, they told that there are invisible beings which are causing these organ infectious diseases. They didn't know which invisible being, they couldn't see it with their naked eye, but there was some kind of infectious diseases. When this was happening, Anthony Van Hook was a draper, that is, who grinds lenses, who works in the lens shops in Delft of Holland. He had an hobby of grinding the lenses. So when he used to grind the lenses, he observed various things. And while doing such experiment, he found out three major morphological forms of bacteria. So he communicated all these bacteria that was seen under those grinded lens. He told that there was some kind of bacteria which used to cause infection to the Royal Society of London. So his observation had caused a major impact on the study of microbiology. There were many other scientists who were very much interested in Anton Leeuwenhoek's simple microscope. So they started discovering various other forms to observe the microscope. So the next contribution, which we can see here, is Agostino Bassi, who showed the pathogenic role of microorganism with Muscardine disease of silkworms caused by fungus. So Agostino Bassi, he observed the Muscardine disease that was seen in the silkworms, which was caused by the fungus. Then we will see about Oliver Wendell Holmes. He is also a famous scientist who observed the purpureal sepsis, which was transmitted by contaminated hands of the obstetricians, that is the people who treat patients, that is doctors. So Oliver Wendell Holmes and Ignace Semmelweis together, with not together, they were independently working on this purpureal sepsis which was happening in their hospitals. So to prevent this, they advised them to use antiseptic solution to wash their hands. So that is how they prevented the contamination that and the sepsis of the wound. Now we have Nadim, the Irish priest who believed in spontaneous generation of microbes from the decaying matter. Nadim was a priest. He believed that the spontaneous generation, spontaneous is there wasn't any other microorganism which was reproducing into another microbe. It was a spontaneous one. It is a self-triggered, that is called abiogenesis of the microorganism was being observed in the decaying matter. He put a decaying matter, decomposing organic matter on petri dish and observed that there was a spontaneous generation. The microbes growed themselves. He told this experiment and told that it was because of spontaneous generation. So then we have Spallanzani, who okay, had opposed the view of Nadim because he didn't agree with Nadim's view that the microorganism spontaneously generated was not acceptable form. So Spallanzani found out that there was no such a generation of microorganism after long heating of petroleum fluids. So petroleum fluids are the ones which are very convenient for the microbes to grow up. So Spallanzani found out that there was no microbes growing after a long heating. So he proved that the organic contents of the flask supported the microbial growth when exposed to air. He proved that only on exposure to air the microorganism started to grow and not because of the spontaneous generation of it. Then later we have Pasteur, one of the famous microbiologists also. So Pasteur proved conclusively that all forms of life, even microbes, arose from their like and not de novo. So, according to Pasteur, it was that all forms of life, whether it's microbe or human, it arose from its own like. That is, microbe arose from another microbe, and human arose from another human. So that is how the forms or the forms of life started reproducing.